Hi and welcome to my second video in the series about membranes, and today we're going into further detail about the plasma membrane. So last time we finished by looking at the phospholipid bilayer, and today we're going to look more into detail about some of the proteins and some of the things that are found within the plasma membrane. So a phrase you may have heard used to describe the plasma membrane is the fluid mosaic model, and the reason for this usage of the term fluid is because the proteins and some of the ion channels that actually sit in the plasma membrane are able to freely move around, hence the phrase fluid. The ease at which they do this is dependent on the number of phospholipids with unsaturated fatty acids. So an unsaturated fatty acid is one that doesn't have its full valency full up. And so as you can see, some of the plasma proteins that are in the phospholipid bilayer are free to move around. So what we have here is a diagram of a plasma membrane, and it contains a plasma protein, something that spans across the phospholipid bilayer. So this section at the top here is the extracellular space, the outside of the cell, and this section here is the cytoplasm, so the inside of the cell. You'll notice that these yellow things here, which are fat-soluble molecules, so they're able to dissolve into fats, are able to freely move across the lipid bilayer. As the lipid bilayer is made of lipids, therefore it enables fat-soluble molecules to move across more easily. Whereas polar molecules, these are usually charged molecules, things like ions, they require a pore, they require a protein channel in order to move them through, and hence you can see one moving through there. So of the different types of proteins found in the membrane, here are just a few. You have this section here which is called glycocalyx, and that comprises of a glycoprotein and a carb chain. They enable recognition of cell tissue. So for instance, if you have a group of cells that you want to bunch together to form a tissue, these structures here, they will enable recognition. We've got peripheral proteins and integral proteins. Sometimes the integral proteins are known as intrinsic, and the peripheral proteins are sometimes known as extrinsic. You also have a carrier protein here, these two red sections here, and a hydrophilic channel. So anything that doesn't like water can travel through this section. Like I said before, usually chemicals, or usually things like ions. So here we have some examples of carrier proteins found within the membrane. So not all of the proteins are used for recognition. Some are used to move things from out to inside, or inside to out of the cell. And so there are different types here. You have a carrier protein to do with passive movement. You also have a gated channel protein, other types of channel proteins, some which actually require active transport. And in the next video, that's what we're going to be looking at, the different types of movements across cell membranes. So in summary, we have proteins that enable to move throughout the plasma membrane, and hence the phrase the fluid mosaic model, because the proteins are allowed to move, hence the word fluid. There are peripheral proteins found on the inner or outer face of the membrane. They're sometimes referred to as extrinsic. Integral proteins found through both lipid layers, and hence they're sometimes called intrinsic. Glycoproteins form the glycocalyx, and these allow cell recognition. Receptor proteins can act as binding sites for hormones and other substances, and can transmit the information to the interior of a cell. The variety of carrier proteins allow for controlled movement of substances through the membrane using passive diffusion or active transport, which is what we're going to look at next video. Non-polar lipid-soluble molecules can diffuse through the phospholipid bilayer. Ionic polar molecules, they require a carrier protein to enable them to pass through the membrane. If you join me next time, we'll be looking at active transport, facilitated diffusion, and diffusion itself.